Hello folks, welcome to the first video, all about inheritance. I'm going to do some worked examples with these and then there will be another example over the page that you can pause the video and try yourself. We're going to start really simple and a lot of what we talk about will be familiar, but the idea is that we're going to build up to the more complex problems. So the very first thing I'm going to talk about is something called a monohybrid cross. It's something that you might well have done at GCSE, so you're very familiar with it. But I'm actually going to start with real basics. This is the problem we're going to solve in a minute, but before we get into that, I want to talk a little bit about the fundamental idea that you have to have in your mind when you're doing inheritance problems. They're usually about two parents having a child and working out the probability of the child inheriting a certain condition. But in order to work those out, you've really got to be clear in your mind what is going on when we produce gametes. Inheritance is all about gamete production. And if you can be really clear at this stage, and then continue to be clear when we do the more complex problems later on, then that will make it easier. Now, a gamete is produced from a body cell. Here's a body cell. There are going to be some chromosomes in it, and let's take our example where we had a heterozygote. Hetero means different, and we were talking about this genotype here, DD. So this is for the dimples example. Now, what does that actually mean, heterozygote? It means that they're different, and those are going to be found on a chromosome. Now there's two letters because in a body cell you will be diploid. Now I know that for a human body cell there's actually going to be tons of other chromosomes in there. There's going to be X and Y maybe. There will be some other autosomal ones in there. Let's put a small chromosome, crumb that in that corner there, and maybe another one up here. But the good thing about inheritance problems is that, yes, I know there's a ton of chromosomes in there, but you can actually disregard all of the ones that don't have the gene you're interested in. So we're not interested in X and Y. We can ignore those. These two pairs of chromosomes here, let's get rid of those. They haven't got the gene we're interested in. So we're just focusing on one pair of chromosomes. Now I've done a blue and a red here. The chromosomes are the same size. They contain the same genes. But you will have inherited one of these from your father and one of these from your mother. Which is why I've done them as different colours. Now that can be important later on. But it's worth remembering that chromosomes come in pairs in what we'd call diploid cells. Now we're going to go through a bit of meiosis. So we know that the first thing that will happen is that these will condense and you'll see the chromosomes becoming visible. But then in metaphase 1, they will line up down the equator. And in fact they're going to line up in a double line. So if the other chromosomes, if we were still looking at them, you'd see that they are also lined up like this as well. We're not interested in them, so we can ignore them for now. But this is important because it means that we're going to go through our first cell division. So the cell is going to elongate like this. The chromosomes are going to be pulled to opposite poles by those spindle fibers, remember those? And then ultimately we're going to get the cells dividing in our first division. And we've gone from having two of those genes, two copies of that gene in each cell, to just one. So at this stage they are indeed haploid. The genotype of this cell is D and the genotype of this cell is capital D. Now I know, and you know, that of course meiosis has two divisions. So yes, in theory these are sister chromatids that could be separated out again, but look, those sister chromatids are identical. So in inheritance problems, although there is another division, you kind of stop here. Yes, this one is also made of sister chromatids, but look, they are identical. For the purposes of this exercise, we can say, right, we're done. We've made gametes with these genotypes. Now, how does that play into our example? Well, I've just shown you the gametes that can be produced. So we've got father up here. He's a heterozygote, so he will have dimples. And we've got the mother here, also heterozygous, so she's got dimples too. And I've just shown you how you would produce those gametes. Okay, so there were their parental genotypes. We're now going to make sperm cells that will have either a capital D or a little d in it, and egg cells will have either a capital D or a lowercase d. And you've probably seen these diagrams done before. You're showing what happens when those fuse together. Okay, you're now going to make a fertilized egg, a zygote that has got two dominant alleles. This one is going to be dominant and a recessive, this one's going to be dominant and recessive, and this is going to be 2 recessive traits. Now it says, what is the probability that the child has dimples? You always have to answer the question. Think about which ones have got dimples. Dimples is caused by this dominant gene, so that one's got dimples, that one's got dimples, that one's got dimples. That is 75% probability. So always relate it back to those gametes. Okay, that's the first one. Pause the video here and then have a go at this example that's on the screen. Sickle cell anemia is caused by a recessive allele. Show how 2 healthy individuals can produce a child with the condition. Have a go at that one yourself. Right, here's you checking that your working is a bit like mine. So this one doesn't actually give you the letters, so I'm going to go with S's, okay? Sickle cell anemia, that's the healthy version there. And we want 2 healthy individuals. So both of our individuals need to have at least 1 copy of that. So I'm going to have 1 parental genotype there. I'm going to cross it with this parental genotype here. Now they're going to produce a child with the condition. So they both need to have the recessive allele there. So if this is our cross, these are our 2 parental genotypes. The gametes we're going to produce are like that. Just notice I'm trying to make my big S's and little s's really different so that there is no confusion. And I'll move that slightly, there we go. You can see that this is the sufferer here. There's a 25% chance of getting a child with the condition. Alright, hopefully you arrived at something similar yourselves.